I'm going to mute for the time being.
Okay.
You can mute until I hand it over to you, I guess. Okay, I think we're going to get started. Hopefully everyone is able to follow the link and get in okay. Hello and welcome, everyone. If there's anyone here that attended last week's Journal Club, welcome back. For everyone else, my name is Jonathan Nasi, and I'm the Director of Translational Science here at Inscopix. Our Journal Club series is a new effort to help keep the scientific community connected during these difficult times. We'll be periodically discussing publications of importance to the field of circuit neuroscience, often featuring Inscopix, Invista, or Invoke imaging, and we hope that you can join the discussion from your virtual offices at home. You can send us questions via the chat window throughout the presentation and for up to 5 minutes after the presentation is over. We'll do our best to get to most or all of the questions with a hard stop right at 12 p.m. Pacific time. So today our field scientific consultant, Morgana Favero, will be presenting a recent Neuron publication from the Patel lab at Vanderbilt University. Morgana is an MD PhD from Italy and was in fact an assistant professor at the University of Verona for a couple of years before moving to the United States. She first worked as a scientist at the Children's Hospital of Philadelphia Research Institute, where she became deeply immersed in imaging techniques before joining us as a field scientific consultant. Morgana, we're really excited to have you join us today, and I'll hand it over to you now so that you can lead us through this paper. Thanks.
Hello, everyone. Thank you for attending the seminar, and thank you, Giorgio, for the nice introduction. So I will be sharing my screen. Let's see. And so we can get started. Let's see. Okay. Okay, I guess I have to swap it. Okay. Okay. Seems like we are ready to get started. Yes.
Actually, we now see your like presenter view with notes and whatnot. The first time it looked good.
Okay, so I think okay, so let me swap it back. Okay.
It looked good the first time, I think.
Yeah, so, okay, wonderful. Now I don't see myself, but I don't think it's a big deal. Okay, anyway,
You're not sharing at the moment, though. You're not sharing at the moment. Maybe stop sharing, redo it.
Yeah, okay, so share screen, share.
There you go.
Okay.
That looks right.
Okay.
Yep, perfect.
Wonderful. Okay, yeah. So yeah. So the paper that I will presenting today has been recently published in Neuron, and is from Patel lab at Vanderbilt University. And so, of course, the topic is very important because it's known that stress is a major risk for neuropsychiatric disease, specifically for anxiety disorder, major depression, and of course is the cause of the post-traumatic stress disorder. So therefore, it's very important to understand the circuit level mechanism by which stress exposure is translating to distinct pathological behavior. And since this could also have, of course, broad translational implications and find new therapeutic approaches to mitigate the adverse effect of stress in the mental health. So it is, in this context, is very well known that the functional connection between basolateral amygdala and dorsomedial prefrontal cortex in humans has been implicated in generation of negative affective states. The dorsal medial prefrontal cortex in humans has a functional analogous in mouse, and is the prelimbic prefrontal cortex. So the authors performed a series of experiments in mice to show that, first of all, they wanted to to establish and confirm that the stress have an effect on this BLA-PIPFC connection, and particularly strengthen the connections. And then they also did a series of experiments to understand how this connection would change the how stress would affect the BLA-PIPFC connections in neurons that have a reciprocal innervation to the BLA. And also they will show data on how those other neurons connected so that receive projection from the BLA, but they're not projecting back, are affected by stress. Finally, they will show what the important role of endocannabinoids on these BLA-PSC inputs during basal conditions, during stress, and they're going to do this using pharmacological and genetic tools. So the first experiment, series of experiment, shows that confirms basically that the BLA-PFC circuit is stress responsive and anxiogenic. So in the very first experiment, they used fiber photometry to image BLA terminals in prelimbic PFC. So they injected AAV-GCAM6 in the BLA, and then they implanted optic fiber in PFC. So they waited, this is the timeline for the experiments. So they waited several weeks. Then they performed the fiber photometry during a stress paradigm. The stress paradigm in this paper was a series of food shocks, a different intensity, and so the intensity was unpredictable, and it was a series of 20 food shocks. And then after 24 hours from the stress paradigm, they performed the behavioral essays to estimate the anxiety-like behavior in mice. So here the results of fiber photometry shown that there is an increase of fluorescence, time locked with the shock delivery. And this of course confirms that this the fiber photometry was implanted in PFC, so of course it shows that this BLA-PFC pathway was highly activated by stress. In the second experiment, they actually used an Inscopix miniscope to image from the PFC neurons, and of course they could image the single cells. So they injected AAV-GCAMs. The time, the experiment timeline is basically the same that is shown here. So they injected GCAM7 in prefrontal cortex, and then they inserted the green lens, and they waited several weeks, and then they performed the imaging session during this stress paradigm. Here you see that the bulk fluorescence from the whole field of view shows again that there is a huge increase in the fluorescence, the GAMP signal, time lock with the shock. And when they performed the single-cell resolution analysis, they could see that basically they could identify 3 different populations of neurons. The majority of PFC neurons were responding with an excitatory, they were the majority of the neurons have had an excitatory response during to the stress, to the food shock. A percentage of neurons had an inhibitory response, and some neurons were not responsive at all. When they plotted these, the results, since the excitatory component was much more prominent than the inhibitory component, the overall response from the PFC neurons shows that there is a show that the stress engages this BLA-PIPFC circuit, enhancing the activity of these PFC neurons. So the next step was to perform a behavioral paradigm 24 hours after stress to see how this food shock would affect the in a short time window the anxiety component in the mice behavior. To do this, they used elevated zero maze, which is a quite common behavioral task to estimate anxiety in rodents. And basically, the more anxious is a mouse, the less time it will spend in the open arms of this circle, but it would rather spend more time in the closed arms. So you see here the results, and basically this it shows that the stressed mouse spend significantly less time in the open arms. They also engaged, they tried to enter less times in the open arms with respect to the non-stressed animals. And also they tend to be, to move much less, which is also an indication that they exhibit more anxiety. So the next experiment was they did was to confirm, to try to replicate this BLA-PIPFC activation using a chemogenetics and see if they could have the same results that the food shock was giving. So they used the GQM cherry injected in BLA and a retro AAV3 to activate the selectively the PFC neurons. So they validated in vitro their preparation. So they saw that the PFC cells would increase their actually the BLA cells would increase their excitability during the CNO application. And then basically they performed the behavioral experiments after in 2 groups of mice, the control mice that had only had cherry, and the mice that had the GQ dreads after CNO administration. And here you can see that is the same behavioral experiment, and you can see that the GQ mice spend less time in the open arms, and again they move less. So this was a good is a good experiment to show that basically we can replicate with a dread technology the same behavior that was elicited by stress. They also performed a CFOS analysis and showed that both in BLA and prelimbic cortex, there was an increase of CFOS active cells. It shows that this pathway is highly active during a stress paradigm, or in this case, during DRES activation. So the next experiment was aimed to examine the stress-induced synaptic adaptation within the BLA-PIPFC circuit. So to do this, they injected the mice with a chanurotidine in BLA and also a retrograde AAV TD tomato. So in this way, several weeks after the injections, the retrograde TD tomato would label in red the cells that makes recurrent synapses with the BLA, and of course the chanurotidine could be used to activate the terminals in the PFC acute slices. So this is this type of approach will be used for this experiment and for the next series of experiments. Is also worth mentioning that they were targeting layer 2-3 in PFC. They have several control experiments where they were targeting also cells in layer 5, and they didn't, in layer 5, they didn't see the results that I'm about to show you. So they focused their experiment in layer 2-3. So this, here you can see that again, they performed the stress paradigm on 2 groups of mice, and then so in one group of mice, and they had a control group of mice, a control group, and then 24 hours after the stress paradigm, they prepared acute slices, and here you can see that they measured the EPSC amplitude, and here you see that the stressed animals had a higher, on average, they have bigger EPSC. The pair pulse ratio also shows that is decreased in animals that were stressed. A decrease in pair pulse ratio is an indication of an increased glutamate release probability for if someone is not super familiar with electrophysiology parameters. And also, of course, one increase in EPSC is also shows that there is like a higher synaptic strength. Also, the spike probability was increased in mice that had underwent the stress paradigm, as you can see here, and this was true for most of the intensity that they tried. And this is also another indication that there is increased synaptic strength and an increased glutamate release probability. Then they did the same. They repeated the same experiments in neurons that were receiving inputs from the BLA, so they could be activated optogenetically, but they weren't projecting back to the BLA. And basically, in this case, no difference were found between the 2 groups of animals, the ones that were the stressed one and the non-stressed ones. So of course, as I said, I don't present here a series of control experiments, but I can mention that they replicated the same type of experiments using the projection from the mediodorsal thalamus rather than the amygdala. And they showed that there was no specific changes for this circuit. And this was to demonstrate that this is a specific change that happens in the BLA-PIPFC circuit rather than like a general change that happens every time the mouse is receiving this food shock and express and start to develop this anxiety-like behavior. So it is also worth mentioning that all these experiments were done 24 hours after the stress section, and the effect were still present 3 days after, and also this is also in the is somewhere in the supplements, and or is just mentioned, and then it disappeared 10 days after the stress section. So it means that it's a persistent effect, but it's not a permanent effect. So, of course, the next question that the author asked was to what was the mechanism, how the mechanism that leads to this increased BLA-PIPFC glutamatergic transmission. And they thought that a good candidate to explain this was a change in the endocannabinoid system. So, and before going to into the details of their experiments, I think it's worth reminding everybody how the endocannabinoids work, and especially how they work in this BLA-PIPFC connection. So this is the presynaptic term. So this is a schema that I hope is clear enough. So this is the presynaptic terminal from the BLA, and when an action potential arrives, there is a calcium influx that determines the glutamate release in the synaptic clefts. This glutamate activates the postsynaptic cells, in this case the PFC, and this enzyme, the diacylglycerol lipase alpha, is activity dependent, and is producing 2-AG, which is an endocannabinoid. These endocannabinoids will activate this CB1 receptor in the presynaptic terminal, and this activation will lead to a reduced calcium permeability. So as a result of this reduced calcium permeability, less glutamate will be released in the synaptic cleft. This is a very general way in which endocannabinoids work, and this is true also for this specific circuit. And this it goes by the name of depolarization induced suppression of excitation, or DSE. Basically, is a way in which we might say very generally speaking that synapses are like making more stable. It's also worth mentioning, because they're using this paradigm quite often, that the DSE can be reproduced in vitro. So if in vitro we artificially depolarize the postsynaptic cells, like from a membrane potential like resting around -70, -80, we go to -30. We cause a massive release of 2-AG that will temporarily reduce the EPSC evoked by the presynaptic cell. This will be more clear when I go into their experiments. So this is the first. The first experiment that they wanted, of course, to perform was to confirm that the endocannabinoid signaling in naive mice in wild type was inhibiting the glutamatergic input from the BLA to the PFC. So the first row shows the results when they were recording from cells reciprocally innervating the BLA, and this row is from cells that weren't reciprocally innervating the BLA. As you see here on the top, the first the first experiment, basically, they are evoking the EPSC and see how this CP55940, which is a CB1 receptor agonist, changes the amplitude of the PSC. And here you see that the effect is that it is decreasing the PFC because it's mimicking the 2AG action. And this is true for both populations of neurons. Yeah, and this, so here they, as I just mentioned, they were, they performed a in vitro DSE. So they depolarized the neuron for, I think, 10 seconds. And then they measured the EPSC right before and right after the depolarization step. Here you see that in the vehicle, there is the ratio between, this is the ratio between the EPSC after, the post, and the pre. So you can see here. So the post is much smaller. And when they're applying the CB1 antagonist, or this is DO34 is an inhibitor of the LGL, which is the enzyme that generates 2AG. So in these two, they basically, the net effect is that they are inhibiting the CB circuit. The DSC is basically absent, so the ratio is almost 1. In this, significantly different than the vehicle. And then they also had like a pair post ratio data where they show basically that both CB1 antagonist and antagonist and the inhibitor of the enzyme, they have an effect in reducing the pair pulse ratio. So in other words, it means that they are increasing the glutamatergic transmission between pre and post-synaptic cell. This was true also for for this population of cells that are not making synapses with the BLA, even though probably the results were less prominent, but on average they obtained the same results. I don't go into the details because we still have quite a few slides. So I don't know, Joshua, are there any questions I forgot to ask you?
There's a few here and there, but they're not for lack of clarity. So I think we can continue on. You're definitely presenting very clearly so far. So yeah, I think we can cover these towards the end.
Okay, thank you.
Yeah, thanks.
So I drink also something.
Yeah, absolutely.
It's sparkling water, even though it looks like a beer. So, okay, so this experiment, so this in this experiment, basically what they did was just to confirm how endocannabinoids work in this circuit. And let me take a step back because I forgot to mention this this actor in this whole story, and it's just important because they're gonna use also some inhibitor of this MIA-GL, which is a monoacylglycerol lipase, which is responsible for the degradation of 2AG from 2AG to AA glycerol. So this is important because it's gonna come out in this slide. So so what they did here was to try to see if the stress was impairing the 2AG inhibition from the BLA in the BLA-PIPFC circuit. Okay, so here they used, they have these groups. So the non-stressed mouse, and they use mass spectrometry to measure the 2AG and also this AA, which is a catabolist. So they show that basically in the stressed animals there is less 2-AG and less AA. And then they used this JZL184, which is an inhibitor of the enzyme that is responsible for degrading this 2-AG. So the overall effect is that it's going to increase the levels of 2-AG. And if you see here, actually, you can see that when they use this inhibitor, there is more 2-AG. But and then there is less AA here. So this is important because it suggests that whatever is happening during stress is not a reduction of inhibition, is not an increase of MAGL action, but is more like an increased production of the 2-AG in the postsynaptic cell. They performed the elevated zero maze again, and they show that of course, they confirmed the difference between non-stressed and then stressed animals between in the open arm time and entries. And then they show that this effect is reversed, is rescued by the increased level of 2-AG. And the situation is impaired when they're using an inhibitor of the LGL that basically has the effect of reducing the 2-AG. So these are all very good indications that stress itself impairs the this action, this inhibitory action of 2-AG in this BLA-PIPFC reciprocal circuit. Then they performed this, basically the same type of in vitro experiments that we already went through in this new condition. So using these inhibitors. And so here the left side is for the reciprocal inhibiting cells, and here is for the PFC cells that are projecting, they're not projecting back to the BLA. So, so here they basically, in the pair-pulse ratio, they find that again, they confirm the previous data. Then when they use the AGL inhibitor, they found that this group behaves as a stressed group. So this is the non-stressed, right? So it goes down because there is less 2-AG. But it's also interesting to notice that this occludes the effect of stress. So there is no difference between stress and non-stress in this group. So it's interesting because it means that the pathway that leads to this result might be the same if there is occlusion. And then when they use this, they increase the level of 2-AG, they rescue basically the effect of stress. So there is no difference between these two groups neither. What time is it? Okay, so the pair-pulse ratio and the, which is, oh, this is the pair-pulse ratio. So the DSA experiments basically goes in the same direction. So confirming that this 2-AG is a good candidate to mediate the stress-induced increased glutamate transmission. And interestingly, in the cell population that is not in the PFC that is not projecting back to the BLA, there is basically there is very little effect on some experiments confirm this population of cells, but in other like the DSA doesn't show a lot of difference anyway. So the overall so in this cartoon they propose a model where they show how the reciprocal BLA and PFC synapses are strengthened during stress. So here, this is a basal basal conditions. So there is 2-AG released by all these neurons, and then during stress, this pathway specifically is in this pathway the 2-AG is reduced, and these, of course, these neurons projects back to the amygdala, and they enhance also the amygdalar connections to ventral hippocampus and central amygdala that are very important brain areas to involved in anxiety behaviors. Okay, so after the pharmacology, the authors also they performed a couple of very elegant experiments using genetic strategies. So the first experiment was done using this DAGL mouse, which knockout that was generated by the same group and was published a few years ago. I don't remember exactly the year, but basically is a mouse generated by this group. And so what they did here was injecting. So this is a conditional knockout. So they injected AAV-CRE in the PFC, retro AAV-CRE in the PFC. No, sorry, it's not retro. It's an AAV-CRE in the PFC because they want basically to delete this the AGL in the prelimic PFC specifically, and only in PFC. And then they injected AAV to the tomato and channelrhodopsin in the BLA. As a result, so here they validated with histology, and it's not probably extremely clear on the screen, but it shows that in the prelimbic cortex specifically, there is no DAGL expressed, while is expressed in the infralimbic cortex. So they really aimed to have a very selective deletion of this enzyme to make sure that they were revealing a very specific mechanism rather than a generalized absence of DAGL in other parts of the brain. Okay, so then they did electrophysiology again, and they show that the purpose ratio, so show that there is the knockout DLGL shows that there is a decreased ratio, suggesting that there is an increased glutamate release, and also the DSC experiment shows basically that there is much less pronounced in mice that don't have DAGL. In neurons, in neurons that don't have DAGL, so they don't produce 2-AG. They also using the same mice. I mean, it's different the group, but the same genetic strategy. They performed the behavioral experiment, and they showed that of course the CRE mice behave. They have a more pronounced anxiety behavior than the GFP mice, and this is enhanced by stress. So, of course, stress brings down also the control group, because this is what stress does. We've seen like at the beginning of this presentation. But if you see the scale here, so it means that it shows that the deletion of the AGL, and so the depletion of 2-AG is able to enhance this anxiety behavior in mice. And here, this is a scheme of how to explain the results of the experiments. So in this case, there is no DAGL at both both populations of neurons, they projecting back to the amygdala and the ones that don't project back, because the enzyme was basically removed from all the PFC, prelimbic PFC. Okay, so the last experiment is they also use another genetic strategy, which is called Intersect, and it has been published. If somebody's interested in the details, there is the construct is in the supplements, and also it has been published by Desserot Lab a few years ago. By the way, this the lab generated a mouse where they could selectively delete the CB1 receptor from specific brain areas. Basically, the CB1 gene was flanked by 2 LOX-P sites, and then so if you see. Sorry, I forgot for all the time that I had a pointer. Giorgio, you could tell me. Forgot it. So anyway, so the so here you can see that what they did was to inject retrograde flip TD tomato in the prefrontal cortex, and this would selectively delete the CB1 receptor in the BLA cells projecting to the PFC. And then they injected sorry, kind of lost here. So, okay, so the retroflip recombinase in the prefrontal cortex, and then in the amygdala, they injected this, okay, they need a CRE, AAV CRE in the amygdala to delete, to have the selective deletion. And then they also injected the channelrhodopsin as the same as before. So the overall results of these strategies that basically the BLA projecting neurons in PFC lack CB1 receptor. So here you see the results of these experiments. Again, they use the same CB1 receptor agonist, and they show that there is no effect of the agonist in the CB1 knockout mice, basically. So they want to confirm. They wanted to confirm that this strategy worked. While in the wild type there is a reduction of the EPSC amplitude, and yeah, this is the same data basically represented in as a population data. And also the pair-pose ratio shows that in the CB1 knockout, there is a reduced pair-pose ratio, and also the DSE is way basically a very less pronounced DSE respect to the wild type mice. Okay, so now after confirming with electrophysiology that the preparation worked as expected, they performed histology. They performed the behavioral experiments, and they found that they found interesting that in these conditions, in the basal conditions, there is no difference between the flip mice that don't have CB1 receptors in these BLA projecting neurons and the control TD tomato. But then when after the stress paradigm, this difference between the two groups comes out. And so the explanation for these different behaviors is that since this CB1 deletion is very, very specific, it's very possible that in a basal situation, the CB1 endocannabinoid systems that coming in the prefrontal cortex, but also in general, is basically how to say, is enough to maintain the behavior, the animal behavior, as in the same range of the control animals. And okay, and this is also the final explanation for this, the representation for this final experiment. So in this condition, these cells lack of CB1 receptor. So when there is in a stress situation, there is still 2-AG produced by the postsynaptic cells, but the lack of receptor in the amygdala makes these amygdalar cells more responsive and more likely to change their connectivity also with other brain areas important for the anxiety behavior. I think I'm almost in time because I have a last slides where basically just want to summarize a few the most important results of this paper. So the author confirms first that the stress causes selective strengthening of synaptic transmission in this circuit BLA-PIPFC BLA. This increased glutamate transmission is driven by the collapse of retrograde 2-AG mediated endocannabinoid signal. And okay, so I didn't really mention, but the DSC is an expression of phasic inhibition, and the pair-pulse ratio is an expression of tonic inhibition. It's a very subtle difference. I mean, it's not subtle, but I didn't really talk about this during my presentation, so we can skip for now. So the single cell these last 2 points are more like a general consideration that the single cell calcium imaging was done in the neurons in the prelimbic prefrontal cortex, and it suggested there was a broad activation of those neurons. So of course, it might be interesting to do more like specific experiments also, and also maybe try to perform like experiment in a more longitudinal way to see how this cell population might change during time after the stress administration. And also it's also interesting to mention that this specific circuit is looks like a very good biomarker for novel therapeutic strategies, and mice can definitely be used for a good preclinical studies, and find a new strategy to of course not to relieve some of the mental disease caused by increased anxiety or increased stress and increased anxiety. So I think I'm done. So we have, yeah, I was just in time. So 44.
Perfect timing.
Yeah.
Perfect timing.
Yeah. So I skipped a few things here and there, but I guess.
Yeah, we didn't go through the whole supplementary material. I was expecting, I'm just kidding.
Yeah, sure.
No, that's okay.
Well, I mentioned here and there where I thought it was necessary. Maybe that they did. Of course, they did a lot of control experiments, and
right.
Yeah. So if someone is, I was thinking maybe someone might have the questions, so I can go through.
Right.
Questions. Yeah.
Sure. Yeah. No, thanks, Morgana. I really appreciate it. You went through that really clearly. Honestly, for me, with all of the ex vivo sliced ephys and pharmacology, it's actually not that easy to go through all of that from my perspective, but you did a really nice job, so I appreciate that. Yeah, and we do have a few questions here. One is, you sort of hit on this at the end actually, but one thing was just actually going back, maybe you can actually go back to figure 1 in your slide deck. But yeah, I mean, I think that, I mean, obviously they did an amazing job really probing in detail the endocannabinoid signaling cascade and its involvement and relationship with stress in this particular circuit. But it seems like, you know, there's quite a few things that I was certainly left kind of really interested to probe further in terms of the in vivo imaging, for instance. And you sort of hit on this a little bit, but one is that so I guess they use a synapsin promoter here, and they're just broadly imaging from the cells. And they mentioned that almost half of the cells, it looks like, I mean, there's a net positive increase, but almost half of the cells are increasing their activity, and almost half are showing suppressed activity. So I mean, it really just, yeah, it left me kind of really wondering, you know, who are those cells, you know, is there any, they didn't really touch on this. I was kind of curious if there was any spatial organization to the activity that they saw, or any laminar distribution. When you target prelimbic cortex with the Grin lens, typically you're coming at a orientation that allows you actually to potentially span layers as well, and pull out some of that information. So, and then of course, you know, they use all these really fancy genetic tricks, which was pretty remarkable. And I'd love to see some of those applied, you know, to the imaging as well. Obviously, you know, it would be feasible to record, you know, specifically from the BLA projecting prelimbic neurons. Yeah, so I don't know, I guess I was, you know, it's not exactly a full question per se, but like, I'm curious your thoughts about what you all kind of felt would be interesting follow-up. It's always easy to point out what they didn't do when they did so much, but yeah, it might be interesting to comment further on that.
Yeah, actually, I was also hoping we should ask them, right, more than me, because I don't, maybe they're already doing those experiments and we don't know, but yeah, actually, your point is very good. And so they were targeting the prelimbic cortex, but I was also surprised by these results. And I mean, not surprised, because as you said, I actually double check, is they don't say, yeah, it's a GCAMP7F, but it's a pan-neuronal virus, so there is no any. So of course, the first thing it might be interesting to do is definitely, I would at least maybe limiting to layer 2, 3, for example, using one of those genetic lines that, you know, because we've seen, so I briefly mentioned that they did the same, they did a lot of recordings in layer 5, and they didn't find any difference. So and then another thing is, of course, they use all those very nice genetic tools at the end, so they might just try to maybe to target specific, of course, but first of all, they might just try to target those reciprocal innervating cells. That would be great. But also it might be very interesting. So here they have data only on the stress paradigm. And then it would be very nice to, of course, to image during the behavioral session, like the open maze, or this open zero maze. Sorry, is zero, whatever.
Yeah, they use a couple.
Yeah. Anyway, yeah. So they also have a T maze, but most of the times they use this one. Anyway, and also I also mentioned that they have they try to do the behavior 24 hours, and then 3 days and 10 days after the stress, and they found that 3 days was still there. I mean, there was some behavioral changes, and then 10 days was disappeared. But of course, it might be very interesting to see what those neurons are doing during this time.
Yeah, I completely agree. And I think in particular, since most of the ex vivo work was done probably 24 hours after, it would have been really nice to do some in vivo imaging during that time period to see what's going on during that behavior. Yeah, and I think just more generally, like you know, like you said, there's this really broad activation of prelimbic cortex during stress. yet the endocannabinoid signaling impairment seems to be much more specific. And so yeah, I think it'll be really interesting to probe into the circuitry there and understand kind of what's happening in between those two things. Yeah, just one other comment really is that kind of along the same lines, but it also occurred to me that in Figure 2, which I found to be a really nice approach in general to
this one.
Yeah, Figure 2, I believe, or no.
Figure 2. Yeah.
Yeah. Yeah. Yeah, this one here, just the general approach that they took to be able to, record in slice from the BLA projecting or BLA, not projecting cells in prelimbic, and optogenetically stimulating the BLA inputs, I think was a really nice approach. And I thought, again, obviously always a little more challenging in vivo, but it seems like, in fact, with our Invoke miniscopes, one could start to do these types of similar experiments. I mean, I was thinking that if one were to inject like an AAV, C1V1 into BLA, and then image in the PFC, and label neurons in the PFC retrogradely from the BLA, right? So you're expressing GCAM just in the BLA projecting cells. You have the fibers that you can activate. Yeah, it seems like that would be another nice potential follow-up
Yeah.
these studies to do in vivo.
Well, maybe they're doing that, right?
Yeah, no, I mean, probably
Yeah.
that's coming soon,
Yeah, so we just have to wait.
Yeah, exactly.
And yeah, but I saw the same thing, that this optogenetic approach might be very very nice to be done in vivo experiments. Yeah. With calcium imaging.
Yeah, and the only other question or comment that came in was just that clearly this is a really active area of research, and really a lot of interest translationally for development of therapeutics that might target this endocannabinoid pathway in the context of stress. And so yeah, it seems like another thing that will be really interesting to see would be starting to study these same things that they're studying here, but in maybe more pathological contexts, whether it's, in models of actual, generalized anxiety, PTSD, things like that. Here, it's a very acute kind of look at this, and clearly there's involvement of these pathways. But yeah, it'd be nice to start to also look at these in, more disease kind of models potentially, to see if these same kinds of principles hold up and if there's any differences.
Yeah.
Excellent. Okay, well, I think we're just about to hit noon, and we're supposed to have that hard stop then. So I think we will finish up if there are no more questions. So thanks to Morgana for walking us through this interesting publication. And thanks to everyone else for attending and sending in questions. We hope this was scientifically stimulating for everyone, and that you'll join us again at the same time on Friday next week, when our field scientific consultant Ching will be presenting a new study from the Papaleo lab at the Italian Institute of Technology on the topic of somatostatin interneurons in the prefrontal cortex and their control of affective state discrimination. Should be a worthwhile read. So please stay safe and healthy, and hopefully we can all get back to our regular research activities very soon. Thanks again, Morgana. Really appreciate it. And everyone, take care.
Welcome. Bye-bye.
All right, great. Thanks.